Hi, my name is Stephen Ling, and today I'll be going over a couple of slides for Biology Lab 172. So I'll start with Zygomycota. The life cycle of Zygomycota has a sexual reproduction cycle and an asexual reproduction cycle. Regardless, they both start with spores and end with spores, which are the haploid reproductive cells for these molds. So when these spores land on something that they can grow on, such as a loaf of bread or fruit, they germinate and form hyphae, which are these strand-like structures which get together, clump together, and form mycelium, mycelia, which are essentially the main body of the mold. Eventually, at 3 here, we see that the mycelia sprouts new hyphae with these structures on the end, which are the sporangium. These sporangia produce the spores and help disperse them so that the cycle can begin again. Now, in asexual reproduction, the Zygomycota can afford to do this because there's food nearby and there's nothing threatening the mycelia if it grows there. But in sexual reproduction, it might choose to do this in case there isn't food nearby or something else is keeping the Zygomycota from freely growing. That's because it forms a zygosporangium at the end, which helps protect it and stay dormant until favorable conditions present themselves. So in this sexual reproductive cycle, we have the spores form the hyphae get together, form mycelia. At number 6 here, however, we have two adjacent mycelia with opposite mating pairs. So this means that they form a gametangia, which is shown in number 7. The gametangia is still haploid, that the nuclei are haploid, because if you can see from the colors here, they're still separated by this cell membrane in the middle. Between 7 and let's say 7.5, plasmogamy happens, which as you can see is because that cell membrane separating the middle part has disappeared. So there's an intermingling of plasma. However, the nuclei are still haploid and remain separated. So in 8 here, we see that the zygosporangia is formed. It's got a defensive wall around it, which means that if, like I said before, those conditions for growing are unfavorable, the Zygomycota can essentially just hibernate or stay dormant until it can grow. So while inside this Zygomycota, karyogamy happens, and this results in the diploid nuclei. So eventually, when things are safe, the diploid nuclei undergoes meiosis, which you can see in 9, and which results in the haploid sporangia. And those sporangia produce the spores and the cycle begins anew. So in this microscope slide, we see, okay, this looks like zygosporangia because they're generally darker and generally easier to see. They're less transparent. Like this here might be a sporangia and so might this. But, another key feature is that they have the two opposite mycelia on the sides of them sometimes. This one's a pretty clear picture. So based on this, we can infer that this sample is undergoing sexual reproduction and might be because it just didn't have enough food or something. So next, I will go over the alternation of generation in mosses. So the alternation of generation is something that occurs in plants, and it refers to the fact that during the life cycle of a plant, it'll go through both a gametophyte and a sporophyte phase. So we'll use mosses as an example, bryophytes. So with one, we have this capsule here after the cap has fallen off. That is the tip of the sporophyte, and we'll get to that later. But essentially, these spores are released. Same thing with the molds. These are the haploid reproductive cells. And I guess I'll clear this up now. As you can see from the end result, the top portion of this cycle is the male side and the bottom portion is the female side. So these spores land and you end up with protonemata, which structure-wise, not necessarily function-wise, they are the same as the hyphae, and these sprout, bud, and form the gametophyte. Now, in mosses, the gametophyte is referred to as the dominant phase of the life cycle. Like that's what you generally think of when you think mosses. That's what I think of, like the green fronds that are clumped up on rocks and dirt. And those fronds at the tip of them have paraphyses, which help hold water. And water is necessary for moss because they lack a vascular system to transport water from the ground. And more, also importantly, they need it because the sperm have flagella, and the sperm therefore need water to swim to the egg. But anyways, going back to this gametophyte. At the tip of this gametophyte, in males we have antheridiums, and in females we have archegoniums. Antheridiums produce the spores, and eventually when water hits them, it produces sperm. And when water hits it, those sperm are dispersed. The antheridium holds the egg and just holds it there. It's like a vase and receptacle for the sperm to eventually find its way to. And so that sperm eventually, hopefully, finds the egg and it undergoes fertilization, which up until now, this entire blue portion has been haploid. After fertilizing the egg, we have a diploid zygote. So inside this archegonium, the zygote eventually matures into a young sporophyte, as we can see by this brown tip here. And that sporophyte will eventually mature, and it's got this seta, which is this stalk here, and the capsule, which is enlarged over here. So that is the entire sporophyte. And inside this capsule, which is technically still diploid, meiosis undergoes, the cells undergo meiosis, which results in the haploid spores. Okay, so in this picture, we are looking at the sporophyte, well, and the gametophyte, but so we've got these sporophytes growing from gametophytes or the archegonium. Yeah, the archegonium. And I'm not sure if that's a reflection of the moss, but probably can't see the spores. Anyways, these things will eventually lose these caps, which you can sort of see on this, and that will result in the spores flying to a new location. You can see that the seta helps with the dispersal of the spores, because if they disperse via wind, then they're high up and they can go to places that aren't already settled in. So that is all for my presentation. Thank you.